In today's video, we're going to take a look at the various patterns of inheritance that we study when we look at genetics. So the first one, and the one we've been talking a lot about in the last couple of videos, is known as simple or complete dominance. In the example of pea, you can see that there are green and yellow pea pods, and the capital Y here indicates that the yellow coloration is the dominant phenotype, whereas the green would be the recessive phenotype. Now, if you look at this cross between a homozygous recessive green pea pod and a heterozygous yellow pea pod, we've got some interesting results here. Any of the individual potential offsprings that end up with at least one of those dominant alleles will end up with the yellow or dominant phenotype. Whereas the only way to get the recessive phenotype, which in this case is green, is to be homozygous recessive. So with simple dominance, also known as complete dominance, one allele completely dominates the other. And these are referred to as, or this is referred to as Mendelian inheritance. And it's rather easy to wrap your mind around. Now, this is not always the case for various traits. There are going to be different patterns of inheritance or different ways in which our genes express themselves. And so with skin color, we have something known as polygenic dominance, right? So poly meaning many and genic referring to genes. So our skin or the tone or skin tone is determined by many different genes kind of acting together. And that's why we don't just see, light skin and dark skin, right? You've got a whole wide variety or spectrum of skin tone, right? There's some individuals in the middle, some on the darker end of the spectrum, some on the lighter end of the spectrum. And the same thing can be said about human height. With pea plants, you can say tall or short. Whereas with humans, we've got short, shortest, tall, taller, with most people being around the average phenotype, right? So generally with polygenic dominance, what we see is a bell curve distribution of polygenic phenotypes, right? So this would be extremely tall individuals and really short, but most people falling somewhere in the middle. And we would see the same thing with skin tone. So polygenic is going to control human height, human skin color, and again, going to have a wide variety of phenotypes. Now, we also have incomplete dominance, right? So with Mendel's pea plants, the flowers had complete or simple dominance. They were either purple or they were white, and the alleles for purple overpowered or completely dominated the genes for the white flower. But that's not always the case. Certain flowers, I think like snapdragons, if I'm remembering correctly, they have incomplete dominance, which means that if you have a red flower and a white flower, what we get is pink flowers, right? So incomplete, anytime you think of incomplete, think everything is blending together, right? So if you're like, oh, wait a second, there's an intermediate phenotype or a blending between the two, that's incomplete. Okay, so I think intermediate, incomplete, right? So if there's kind of like a middle mix of the two, that's incomplete dominance. And we can represent this in a Punnett square with two different kinds of symbolic representations, right? Some people still stick with the capital and the lowercase alleles, but you just have to remember that the heterozygous genotype would produce that purple or intermediate phenotype. But I much prefer this. I like to think, if we use different symbols for the different patterns, we won't get them mixed up. So we have R, which would code for red, and R prime, which would code for white. So if an individual has they're just homozygous R, big R, then they only have genes for red. Whereas if an individual is homozygous for R prime, right, the individual might be, R prime, that's the only way to have the white flower. Whereas the pink or intermediate phenotype would be the heterozygous condition. And I know this can be a little overwhelming at first, and that's why I like to use the different symbols to represent the different patterns. So again, with incomplete, just think there's a mixing or blending of the two. And then we've got codominance, right? So codominance, they're both dominating together, kind of like cooperating, you're working together. Codominance means that both genes have equal dominance, right? And so let's say we've got a gene, and let's call that allele or that variation just capital R, and that codes for red fur or red hair, whereas the allele W, capital W, codes for white. And if an individual has both red and white alleles, the animal will be roan, right? And this is just a term used to describe cattle and other animals that have both red and white. So codominance, we see both colors, right? So you'd see like red and white, black and white, and there's not a mixing, right? So this cow is not going to be pink, right? So the red and white are not mixing. You've got red fur and white fur co-dominating these individuals' phenotypes. And so that's why I like to use, again, different symbols for these different patterns. So if you had, right, a red cow, like this one right here, and a white cow, right? And if you cross them, right, if you did a Punnett square, I'll make a little small one right here, right? And this, I think, is pretty intuitive, right? So if we complete this, you know that the only potential phenotypes are going to be the heterozygous roan cows. Now, two roan cows can really produce some interesting variation in their offspring, right? So we can see here in the F2 generation that, you know, these two roan cows, right, they can produce red cows, more roan cows, and white cows as well because they contain both sets or both alleles, right? So this individual cow would be red, this one would be roan, this one would also be roan, and then this cow would be white, right? So there's a 25% chance, right, that two roan cows will give birth to a red calf, and there's a 50% chance that they'll give birth to two roan or a roan calf, and then a 25% chance of producing a white cow, right? So that's codominance, where both phenotypes are displayed in the heterozygous condition, right? They have to have both of the alleles. And let's go on to the next one. Multiple alleles, this is my favorite, and the name implies that we actually have more alleles for the gene, right? So when we talk about alleles, remember alleles are just variations of a particular gene. So with multiple alleles, we've got multiple variations for a single gene. And one of the most common examples that we study in biology would be blood type, right? So there are four main blood types. So these are our phenotypes. So this is what, if you were to look at the blood cell under a really strong microscope, you would be able to recognize these phenotypes. This would be the physical appearance. You have type O blood, type A, type B, and type AB. So we have four different blood types, and these four blood types are the result of a variety of genotypes, okay? Now, here is kind of the odd thing and the difficult thing to remember is that scientists and teachers can use different symbols to represent this, right? So typically I like this representation. I like using these symbols for the alleles. So someone with type O blood is going to be homozygous recessive for O, right? So clearly the genes for type O blood are recessive, right? They're not very powerful per se. Another way you could write this is simply you have a gene for O and then another gene for O, right? Because that lowercase I represents a allele or a variation of a gene that codes for the type O blood type. Now, if someone has type A, right, there's two ways that someone can have type A. They can be homozygous for type A, or they can be heterozygous for type A. So what does that tell us about the allele or the variation for blood type gene for A? It's definitely dominant over the gene for O. So another way I could write this is I could write homozygous for A, right? The individual has two genes for the type A blood type, or they could be heterozygous, and they'd have one gene for A, one gene for O, and the gene for A overpowers the gene for O. And we see the same thing with the B blood type. They can be homozygous, containing two genes for the type B phenotype, or they could be heterozygous. And again, the gene for B, all right, is simply dominant or completely dominant over the gene for the O blood type. So they can be homozygous B, or they can be heterozygous for B, okay? But again, the B would overpower the O, and the A would overpower the O. Now, with the blood type AB, right, you've got a heterozygous condition. Now, this particular blood type represents a codominant example. And this is kind of wild, right? So here we had simple dominance, and now we're seeing codominance because the A nor the B overpowers the other. So this individual represents both blood types simultaneously. Another way you could write this is they have a gene for A and they have a gene for B. Okay. Now, let's take a look at some Punnett squares and try to start interpreting the results because, again, this can be a little overwhelming. So let's look at this first one over here. Here we have a parent and then another parent. Now, the first question is, okay, you can see the genotype, right? But what is their phenotype? What blood type would this person have? Well, A would overpower O, so this person would have type A blood. This would be their blood type. And then this parent, same thing, the B overpowers the O, and this person has type B blood. Now, if someone has type A blood and they reproduce with someone with type B, what are the potential outcomes? Well, this individual right here, right, would have type AB blood, okay? So that would be type AB, okay? Kind of a mix of both parents. Whereas this offspring or this individual would be type B because, again, the gene for B overpowers O. This individual would have type A blood. And then this individual would have type O blood. And this is really quite interesting because blood type is very important when you consider things like blood donors and even organ transplants, right? And so even you could be related, right? You could be the son or daughter of your parents and still have a significantly different blood type and not be a compatible donor to your parent, or you could be the parent of a child and not be a potential donor for your own offspring because of this whole blood type scenario. And let's take a look at a different way of writing this out. So here, this is my preferred way on this side of representing this. I just think it's easier to understand. So we look here, AB. This individual would be type AB. This individual, and this is why I like this, right? Because it kind of reminds us, oh yes, so this is going to overpower the recessive. And so this individual would be type B because that codes for O, but it's hidden by this more dominant gene. And this offspring would be type AB, type B, right? So here we have a homozygous individual for type B. This individual will be heterozygous for A and heterozygous for B. So again, a lot of variety. Now here's a question. With these two parents, is there any potential that they could have given birth to a type O baby? No, I don't see any possibility here, right? And so, blood type could be used as a way of identifying kinship, right? Are you related to your parents? Are you the son or the daughter? But, your blood type could fall into this match and you still, in fact, not be the offspring of your parents because many of us have the same blood type. And that doesn't mean that we're related or that we're brothers, sisters, or cousins. Well, in a sense, we all are, but we'll talk more about that later. But let's look at why blood type is important, right? So, all right, let's start with type A blood. So type A blood is called type A blood because on this surface, we have the type A antigen. These are just like little molecules that stick on the out of the cell membrane of the red blood cells. And we just, we give them a type, right? Type A. And surrounding the blood cells would be antibodies. And if you have type A blood, you have type B antibodies. If you're type B, your blood cells have the type B protein or the molecule sticking around on the outside, and you have the A antibody. So what does this mean? If you have type A blood and you were to somehow accidentally receive a blood transfusion of type B blood, right, your antibodies, right, would attack this type B blood. Right? And this is kind of a good thing. These are kind of a part of our immune system, right? You don't want somebody else's blood inside of your blood. If you think about it from an evolutionary standpoint, there are a lot of diseases that are transmitted from one individual to the next but through blood, right? So naturally there would be, immune system molecules there to destroy any foreign pathogens and anything that's not our own cells. Now, if you have type AB, this is where it gets kind of cool, right? So AB, remember, is codominant, so both of them are going to show this blood cell is covered in both A and B molecules, these antigens, right? Now, because this cell is covered with both A and B, there are no antibodies present. So what does that mean? Well, since there's no antibodies, if you are AB, you could receive a blood transfusion from someone with type B or type A because your body is not going to attack their blood. And so type AB is known as the universal recipient. Okay. Now there are plus and minus RH types, but we're not going to worry about that. You might learn more about that if you take AP biology. And then you've got type O blood. Now type O blood is like naked, right? There's nothing on the outside. And so there's no antigens, right? That's why it's type O. And really, if I remember correctly, I remember reading that type O blood is not actually an O. When people were first studying it, they wrote this, right? Which means like nothing. And someone simply just interpreted that as an O, right? If I remember correctly. But what's neat about type O is although there's no antigens on the outside, there are both antibodies. So type O blood is the universal donor, right? You can, if you have type O blood, you can donate your blood to any of these individuals, right? But the unfortunate thing about having type O blood, because you have both antibodies, the only type of blood that you can receive is more type O blood, right? Because if you give someone with type O AB, their antibodies will attack it, right? So these type O individuals, they have both antibodies. They'll attack these other three blood types, right? Now, what's really interesting about this is you would think because type O is only expressed with the homozygous recessive, you would think that it was the most rare phenotype of all blood types. But we'll take a look at that actually when we get to evolutionary biology and how it's affected the human population. You'll be kind of surprised to see the breakdown or the percentages of how many people have each of these blood types around the world and in our country. And the last one I'm going to talk about is sex-linked. So there are genes that are actually carried on the X chromosome, and they're not found on the Y chromosome. Remember, these are our sex chromosomes. So this individual right here is a male. They have both the X and Y chromosome. And then the female up here has two X chromosomes. Now, the X chromosomes carry specific genes, right? In this case, there's normal blood, and then there's a gene for hemophilia. And hemophilia is a disease that results in a patient or an individual not being able to, there's no coagulation, there's no coagulating agent in their blood. So when they get a cut or a bruise, they just keep bleeding and bleeding and bleeding and bleeding. And it's not a good disease. So this individual right here, this male, is normal, right? Now, how do I know? Well, if I look over here on the X chromosome, I can see the capital H, and that's normal, right? And so we see this capital in lowercase, and you're thinking, oh, okay, it's we got a dominant, we got a recessive situation here. It's completely dominant over the other, but we just have to make sure to pay attention to gender. And then this female up here on her X chromosome, she has a gene for normal blood, and then she has a gene for hemophilia. But since the gene for normal blood is dominant, it'll overpower this gene, and this woman will have perfectly normal blood. However, since we're now studying a disease, we would call her a carrier, right? So she doesn't have the disease, but she has the potential to pass the disease on to the next generation. So if this sperm were to fertilize this egg, what do we get? Well, we get homozygous for normal blood, right? This individual, this female, right, their daughter would have normal blood. And then what if this sperm fertilized this egg? We'd have another daughter, right? We have two X's, and she'd have normal blood because she has that dominant gene for normal blood, but she'd be a carrier. What if this sperm, which contains the Y chromosome, fertilized that egg? We'd have a son with normal blood, right? And then in this situation, if this sperm were to fertilize this egg, here we have a son with hemophilia. Okay, this individual would be affected by the particular gene. And this individual would have to take medications to make sure that they didn't bleed to death. Now, when you look at these questions, you need to be careful. If I were to ask you this, okay, what percent chance do these two individuals have of giving birth to a child with normal blood? Well, the answer would be 75% because this child is normal, normal, and normal, right? They all have normal blood. What if I said, what percent chance do these two parents have of giving birth to a normal daughter? Well, now since I've only asked about the daughter, we're only looking at these two. And this individual has 2 genes for normal blood, and this individual has 2 genes for normal blood, and since the gene for normal blood is overpowering or dominating this gene, these 2 females would be normal, right? So it'd be 100%. Now, what if I said, what percent chance do these 2 parents have of producing an offspring with normal blood, or producing a son? Sorry, what percent chance do these parents have of producing a son with normal blood? Again, now that I've asked about the sons, I'm only looking at the 2 sons, right? And so that would be 50%, right? Because there's only two potential outcomes for the boys, and one of them is normal, and one of them would have hemophilia. So it's 50% chance of having a son with normal blood. So you have to be really careful here when they ask. Is that asking about just all the children in general, or are they asking about just the daughters or just the sons? So why do men are more likely to have hemophilia? Why are they more likely to be colorblind, which is another sex-linked trait? Why are men more likely to suffer from male pattern baldness? And it's because men only have one X chromosome. And so if there's a recessive gene on here, it's going to be expressed. Whereas female individuals, they have 2 X chromosomes. So even if they inherit one of these chromosomes with the faulty gene or the recessive gene, there's still a chance that they might have the normal phenotype because they have 2 X chromosomes. So hemophilia, color blindness, and male pattern baldness are all examples of sex-linked traits.